Hello friends, welcome back to my channel. Hope you all are well and doing good. In my previous video of biotechnology and its principle, I talked about the features of cloning vector, in which I covered oricide, that is your origin of replication and selectable marker, and today I am going to take cloning site, yet another very important feature of cloning vector. So let's start our topic. Now, let's have a quick recap on what we have studied earlier. As we all knew that in recombinant DNA technology, our gene of interest, or you can say a foreign DNA or alien DNA, is inserted into a cloning vector with the help of the enzyme like restriction endonucleases and ligases, which is then inserted into a suitable host where it will get expressed to give us a desired product. Now, the features of cloning vectors are oricide, selectable marker and cloning site. Here in this figure of cloning vector, you can see this feature of cloning vector will represent the cloning oricide, this will represent the selectable marker, and this will represent the cloning site. Oricides or origin of replication are those sequences of DNA from where the replication start, and any piece of the DNA when linked to this site can be made to replicate within the host cell. Selectable marker are those sequences of DNA whose expression will help us to identify and eliminate transformants from the non-transformant ones. Now, the question arises, what is the role of cloning site in the cloning vector? क्वेश्चन अराइज होता है कि क्लोनिंग साइट्स का क्या रोल होता है क्लोनिंग वेक्टर के अंदर. As we all knew that our gene of interest is inserted into the cloning vector with the help of the restriction endonucleases and ligases, but at which site this gene of interest is inserted? Cloning vector के अंदर कहाँ पर हमारा gene of interest जो है वो insert होगा? Here comes the role of cloning site in the cloning vector. Now, let's try to understand it. Cloning sites or restriction sites are those specific sequences of nucleotide on the DNA molecule that are recognized by the restriction endonucleases, and here our gene of interest is inserted. Cloning sites और restriction sites cloning vector के ऊपर वो DNA sequences होते हैं जहाँ पर हमारे restriction endonucleases आकर act करते हैं और यहीं पर हमारा gene of interest insert होता है. Now, let's have a look on the treatment of cloning sites. Cloning sites, or you can say a restriction sites, are located on DNA molecules containing specific sequences of nucleotides which are recognized by restriction enzymes. Multiple cloning sites, or you can say a polylinker. Multiple cloning site is a polylinker, is a short segment of DNA which contain up to 20 restriction sites. Multiple cloning site, as the name suggests, is a short stretch of DNA on the cloning vector that contain a number of cloning sites. Now, the question arises, when we have a cloning site on a cloning vector, then what is the need of multiple cloning site? जब हमारे पास एक क्लोनिंग साइट है क्लोनिंग वेक्टर के ऊपर जहाँ पे हमारा जीन ऑफ इंटरेस्ट इंसर्ट कर सकते हैं, तो हमें क्लोनिंग वेक्टर के अंदर मल्टीपल क्लोनिंग साइट्स की ज़रूरत क्या है? नाउ लेट्स ट्राई टू अंडरस्टैंड इट. Here in this figure of cloning vector, you can see a stretch of DNA molecule has a number of cloning sites and represents the multiple cloning sites or a polylinker. As we all knew that restriction endonucleases will recognize a specific sequence on the DNA molecule. For example, EcoR1 recognize a palindromic sequence GAATTC and makes a cut between G and A on both the top and bottom strands of the DNA molecule, thus leaving an overhang which can bind to its complementary strand and is termed as sticky. And similarly, BAM-H1 will recognize a specific sequence GGA-TCC and makes a cut between G and G on both the top and bottom strands of the DNA molecule. Now, consider a case what happen if all these cloning sites are recognized by the restriction endonucleases. This will result in the generation of the short segments of DNA molecules that no longer hold the property of cloning vector. Now please try to understand this. Although the cloning vector has a number of cloning sites, but at a single time only a single restriction site, or you can say a cloning site, will be active. And for example, if I used EcoR1 restriction site for the insertion of my gene of interest, then all other sites will remain inactive. Now the question arises, then what is the advantage or need of the multiple cloning site in a cloning vector? Now let us try to understand it. Here you can see that there are two figures of cloning vector. Figure 1 will represent a single cloning site in the cloning vector, whereas in figure 2 you can see a multiple cloning site or a polylinker in the cloning vector. As we all knew that in recombinant DNA technology, our gene of interest is isolated from the host DNA with the help of the restriction endonucleases, which is then inserted into a cloning vector, again with the help of restriction endonucleases and ligases, which is then inserted into a suitable host where it will get expressed to give us the desired product. Now consider a case where my gene of interest is located in such a position in the host DNA where I can't use EcoR1 for its isolation and insertion into the cloning vector. In that case, I am not able to carry out my cloning process. But in case of multiple cloning site or a polylinker, I have a number of cloning sites. Although I am not able to use EcoR1 for the isolation and insertion of my gene of interest into the cloning vector, but I can use another restriction enzyme, say BAM-H1, for the isolation and insertion of my gene of interest into the cloning vector, and thus I am able to carry out my cloning process. Thus we can conclude that polylinker will provide cloning sites for DNA molecule by providing number of restriction enzymes into a single plasmid. With this, I am ending this video now. In my upcoming videos, I am going to talk about insertional inactivation and competent host. Please like my videos, share them with your peer group and subscribe to my channel. Till then, bye bye.